Hello friends, now we are going to start with the structure of a DNA. The structure of DNA, that is double helical structure, was proposed by James Watson and Francis Crick in 1953 on the basis of extracrystallographic studies which were provided by the Maurice Wilkins and Rosalind Franklin. What they say, there are several points, there are several characteristics of a DNA double helix structure of a DNA which they propose and which they stated. The first point is nothing but the DNA as a double helix molecule. DNA has a double helix molecule comprises of two long strands. They are coiled around a imaginary common axis, and that forms the double helical structures. Which, if you look at the both the strands, if it is suppose you take an example of a ladder, if you twist it, so the structure which will going to be form is similar like a structure is known as a double helical structure. In this diagram also you can see there are the two strands which are running like from here, like this one, another is one. So both are like this dotted line is nothing but the imaginary axis through around which this both the strands are rotating. They are the one which will be shows the twisting. The second is nothing but the structure of each strand is comprises of nucleotides. In last video also we shared or we discussed about the nucleotides. Nucleotides are nothing but the pentose sugar which will be either ribose. In case of DNA, it is a deoxyribose. Phosphate, nitrogen bases will be the like adenine, thymine, guanine, and cytosine. Uracil will be absent in DNA, that will be present in RNA. So successive nucleotides will be linked by each other. There will be, suppose this is the DNA structure which will be like, suppose this is the DNA structure which is like here over there. Sorry. So this is structure. So this nucleotide and this nucleotides, they both will be linked together by the 3-5- phosphodiester linkage. Means the fifth carbon of the sugar, so fifth carbon of, suppose this is the adenine nucleotide and this is thymine. So for this nucleotide, the fifth carbon which on the fifth carbon there is a phosphate which will be linked with the third hydroxyl of this above one. So they are like the phosphate and hydroxyl. This put two together forms the phosphodiester linkage. That's why it has been saying the phosphate attached to the fifth carbon of the sugar of a nucleotide joined by the third carbon of another nucleotides. Then it will be shows the next point is it shows the complementary base pairing. The nitrogen bases of one strand will be bind to the nitrogen base of another strand. That means there are the nitrogen bases will be the purine and pyrimidine. So purine are nothing but the double ring structure, where the pyrimidine are the single ring structure. Purine always binds with the pyrimidine. So what are the structures which will comes under the in the purine? Purine will be the one like your adenine and guanine. This will be the your purine, where pyrimidine will be the thymine, cytosine, uracil. So there will be a bit confusion how to learn about this, which are the purine and which are the pyrimidine. It's simple. Just focus over here. Just like pare, take care of you. You just take the first initial py for the pyrimidine, T for the thymine, C for the cytosine, and U for the uracil. So pare means pyrimidine, T for thymine, C for cytosine, U for uracil. Pare, take care of you. So once you came, if you just came to know what are the pyrimidine, so then you can other hand you can write it down the which are the purines. Like purines are the double ring structure, adenine and guanine. Adenine always binds with the thymine with the double hydrogen bond, where the guanine binds with the cytosine with the triple hydrogen bond. As I discussed just now, purine and pyrimidine, purine always binds with the pyrimidine. Due to the complementary base pairing, total number of purine always going to equal to total number of pyrimidine, and this is known as Chargaff-Sulz. What Chargaff-Sulz said, adenine and guanine, adenine plus guanine equal to thymine plus cytosine is equal to always number of purine is always equal to number of pyrimidine, or A plus G divided by T plus C is equal to 1. So now we'll come to the another very interesting point. That point is nothing but the polarity. In the last video I also shared with you that each strand comprises of the specific, has come with the specific polarity. That is because of at one end, that is at 3' end, it has a free hydroxyl group, where at the other hand at the 5' end it has a free phosphate group. So one strand runs in the direction of 3' to 5 direction, where the other strand will be runs in the direction of 5' to 3' orientation. So both will be running into anti-parallel to each other. So polarity will be defined by the on third the 3 dash end. If one strand having the 3 dash end as a free end, so that will be the 3 dash end. Hydroxyl at that end, that will be 3 dash end, where the another strand which is having at the 5 another strand or the another end which have a 5 that 5 dash as fifth carbon, it is a free phosphate molecule, so that means the 5 at that has the 5 dash end. So always one end will be having a free hydroxyl group, where other end will be having the free phosphate group, or vice versa. If you look at this structure, it shows this is a deeper groove and there is a this one is the shallow groove. So these deeper and shallow grooves are nothing but the inward projection which has been observed into double helical structure. So when DNA strand undergo the right-handed coiling, that is around the central imaginary axis, it results into formation of a deep groove which is nothing but the inward projection, which will be deeper inside, which is known as major groove, and another is your shallow groove, which is nothing but your minor groove. Just understand the distance between the two major groove. Distance between two major or two minor groove. Always equal to 34 Armstrong unit. The distance between the two major and two minor grooves is always going to be 34 Armstrong unit. So now what about the dimension? The diameter of each DNA molecule is around 2 nanometer. The spider-like arrangement shows the deflection angle of 36 degrees. That means suppose this. So this is the central imaginary axis point, and this is one nitrogen base. Suppose this is thymine and adenine is here. So this, if I draw an angle between this, so this angle will be of 36 degree. So angle will be 36 degree. So deflection of 36 degree between the two successive steps or the rungs. Okay, DNA molecule makes around the one complete day. That means if the distance between two is 36, so it takes 10 molecules or 10 rungs to complete the one turn, that is 360 degree. So after covering a distance of it shows the 3.4, means that is the one major groove and the two minor groove and two major groove completes the one turn completes. So there are 10 base pair which are present into the each spiral into the each circle around 360 degree, that and the distance of the each circle or the distance between two major and two minor is nothing but the 34 Armstrong unit.